Camera 1, camera 2, camera 3. Where's the green screen? Good afternoon, everyone. For those who don't know me, I'm Dr. Joshua Slee. I'm the Division Head of Sciences and Mathematics and the Department Chair of Biology here at DeSales. And it is my pleasure to welcome you to our SURF final presentations. Before I thank a bunch of people, I also just want to give you a little bit of a background of what our SURF program is. Our SURF program is the Summer Undergraduate Research Fellowship in the Natural Sciences. It started actually about 5 years ago as we started planning this program. This is actually only the second time that we've been able to run it due to our building being down, due to HVAC issues, due to that lovely pandemic that we're all emerging from. But we're very excited that this year we had 3 biology faculty. I'll introduce them as they come up for their presentations, and we've had 9 fantastic students. We've had 9 of our DeSales students participate in the summer program, which is a record number, which is great. We're very proud of our 9 students. And the faculty will introduce them as they come up to present. All right. The thank yous here really go out to the university and the administration for their support, both monetarily and in spirit of our program. But then we are also eternally grateful to our Women for DeSales philanthropic group on campus. They have come through for us big for the past 3 years, we'll say, in funding. And they're the reason that we have 9 students here today. So if we can give them all a virtual round of applause and in-person round of applause. So our 9 students this summer, we've basically chained them to the lab. On certain days, we let them out for field trips, but they worked 40 hours a week for 8 weeks straight. We did give them a stipend. We offered them on-campus housing if they choose to take it. And it's been a great summer. They've been working hard, and they're excited to tell you about what they've been doing this summer. Okay. Again, there's a couple people that I want to thank. The 2 most important people I would say are this is Carol Ann Graham and this is Wendy Maybrook. They are the kind of women that run the program for us. So we give them a round of applause. And I see a few VIPs in the audience. We're going to acknowledge Father Jim, our president, who's here today. In the way back, I see Dr. Blumenstein and Brother Dan. So again, I'm going to turn it over to Dr. Alex Krupka, an assistant professor of biology and our neuroscientist. He will introduce his 3 students, and we'll get started. Thank you all.
Hello, everyone. I'm Dr. Krupka. Glad that you are all here. I'm really excited for the students to be able to present what they've been working on. I was originally going to be part of this program last year when it was canceled due to the pandemic. But fortunately, 2 of the students who I was going to invite to be part of my lab, Carl Tice and Ciara Martin, were able to continue this year. So that was great. Also, Kobe Rush, who I know through the Chem Demos program, decided to be able to join my lab, which was excellent. And so I have 3 great students, and the work that they're going to present today might seem a little unusual. We are at a biology presentation. All of us are biologists, and yet you will see maybe not that much biology here. One of the unique things about neuroscience and about all sciences overall is that we are moving to a very integrated, multidisciplinary setup. So what they are going to discuss with you today is the development of a laser system for gently heating an animal, and we can use that to determine when that heat becomes a little painful. You might think of it similar to when you stick your hand under a warm faucet, just when it becomes too much to handle. So Carl, Kobe, Ciara, if you guys can come on up, it's time to begin your presentation.
This is our presentation, the design of a novel laser system for assessing thermal nociception. These are pictures of our rat Rathew. He's very large, very old. My name is Kobe Rush.
I'm Ciara Martin.
I'm Carl Tice.
Okay, so for our research this summer, we had 4 main objectives. The first being affordability. We wanted to create an apparatus that was affordable to test thermal nociception in rodents. In later slides, I will describe what thermal nociception is, but we are just applying a heat stimuli to the rodent's tail. Next, we wanted to create an apparatus that was reliable, specifically for the timing. We were measuring the response from when the rodent flicks his tail. We wanted to create something that was accurate and precise. Previous models have used manual timing, such as a stopwatch or maybe a clock, but we have a new computerized method that is more accurate and consistent. Next, we wanted to create an apparatus that was safe for both the rodent and for the researchers as well. So we want to test thermal nociception without injuring the rodent. So this requires an appropriate wavelength for them. Next, we wanted to have an apparatus that was accessible. So we wanted to create something that people could mimic even if they didn't have any prior experience with a laser. So we wanted to also have something that people could use accessible supplies to make. So why do we make an apparatus that could use for a rodent? So a rodent is a good model organism. First, its genetic makeup is known. It's always helpful when you know the genes of a rodent before you're studying them. Also, they're related to humans. So as you can see on this phylogenetic tree, humans and rats have a similar or a common ancestor. So you can take the conclusions you found from the rodent studies and apply it to the humans. Next, they're cheap and inexpensive to maintain. So they do require daily maintenance, but it's not expensive to maintain their food and water. They're easy to work with. So even though Rathew's a little bit large, rodents in general tend to be smaller, which focuses on one of our goals of affordability. We use less material when we have a smaller rodent. Next, they can be trained easily. So you can train the rodent to stay on the platform by itself without using a restrainer. This will also be an objective of us to have an affordable apparatus because you wouldn't need to make a restrainer, which would save money. So again, as I said, we're testing thermal nociception. Nociception in general just refers to the sensation of pain. There's different types of ways you can test nociception. We were interested in thermal nociception. One of the most famous methods is the von Frey method, and this is to test tactile nociception or pain derived from touch. So essentially it's applied a monofilament, which is like a piece of plastic that has a piece that will poke at the rodent's foot. And the rodent, when it becomes uncomfortable, it will move its hind paw and they can test nociception that way. As I said, we want to do something with heat stimuli. So we went with a laser. However, there are other methods of testing thermal nociception, such as using a light bulb where they shine the light onto the rodent's tail. There is also a hot plate test, which you put water in a beaker, you heat it up, and then you submerge the rodent's tail into the water. Both of these tests are used to measure use for the tail flick test. And this is essentially when the rodent will move its tail out of the way when heat stimuli is present and when it becomes uncomfortable. We went with a laser because our laser will be more accurate and precise. When you have the hot plate test, you're just submerging a random amount of tail in the water. You don't know how much tail is being actually submerged. So this will target a specific pinpoint on the tail, so it will be consistent. And then the tail was just chosen because it's mobile and it's easy to detect. So lasers and nociceptive assessments. As I said, we want to create a wavelength that was suitable for this experiment, not something that was too powerful and that would cause damage to the rodent. When we were reading studies on this, we did find that an 800 to 1,000 nanometer wavelength was appropriate that would create a response but would not be too damaging. We essentially wanted to create a pinprick sensation, but not something that would cause harm. We did test this on Dr. Krupka's arm, and it did create a pinprick sensation, but it was not painful or left any damaging marks, so we believe that it would be the same with the rodent. So now I'm going to get into the methods of our research. So again, we were trying to figure out a wavelength that was suitable. And when we were doing research, we had to take some factors in consideration. Number one, we wanted to create something that was safe, that would not cause pain. Therefore, we had to look at the penetration. So as you have in a larger wavelength, it will penetrate deeper into the skin. We didn't want it to penetrate too deep that it would cause pain. Also, we decided not to go with UV light because it negatively impacts the skin. It can cause skin cancer, liver spots, premature aging, and so forth. Also, with UV light, you can't feel it to respond. We want to measure something that we can see the response time. It's similar to when you're laying in the sun on the beach and you don't really feel the sun hitting you and you don't respond to it, but when you get home, you see that you're sunburned. We needed something where we can test the response. Therefore, IR was the most appropriate to go with. Also, IR light has been used for skin treatments. Sometimes dermatologists use IR treatments. And as well, there was a study saying that Worcester rats were subjected to 890 nanometer wavelength, and they had accelerated wound healing and decreased bacterial levels on the targeted area. So it actually is safe for the rodent and will help them. Again, meeting one of our objectives, we wanted something to be safe. So we did take extra precautions when dealing with this laser. Because it's IR light, you can't see it. It's outside of the visible spectrum, which means if you were unknowingly staring at it for a long period of time, it could cause damage to your eyes. We did get 2 different types of goggles. On the bottom left here, these are from Thor Labs. These are more expensive. And on the top right, we got goggles from Amazon. They're free mascot goggles. So these charts both came with the goggles, and they're just showing the optical density of them. So this has an optical density of about 7, and this is about 5. So with the optical density at 5, it's actually a 0.001 transmission of light. So it's significantly reducing the intensity of the light, making it safe for us to work with. You could go with the bottom left here, but it's not necessarily necessary for our project. And we wanted to create something that was affordable, as I mentioned. So we went with the top right because it is more affordable. This is just a reminder that safety's rad. Always be safe in the lab here. In a minute, Kobe's going to get into the specific design, but again, reiterating, we want something affordable, accessible, safe, and reliable. We also had to create some components to make this apparatus happen. So we needed a testing platform, a restrainer, an easy laser setup so someone can have access and be able to mimic this, and an automated assessment.
Okay, so we wanted to design a whole new apparatus from scratch that fit the goals that CR was talking about. The first thing we needed to design was an experimental platform. This schematic up here, this is one of the early things that we drew. This was drawn weeks and weeks and weeks ago. That's what this design is based off of. So our first design was a Pvc construction. The pros are that it's waterproof, which is really important because when you're working with rodents, they tend to urinate a lot. So you need to be able to clean it. It was really durable. The whole thing was pretty bulky and heavy, and it was relatively inexpensive. The cons are that it was really difficult to work with. Cutting these holes to the right dimensions and precisely was really difficult, especially because the tools that we had were made for wood and not for thick PVC. We needed to build a support frame for it, and the connective pieces were protruding from the surface. The whole thing was pretty bulky and not really what we were looking for, so we wanted to modify this design. Now, our current design, which you may have seen on the back table over there, is made entirely out of wooden screws. It was really easy to work with, since our tools are made for wood, so the drilling and everything was very easy. It was durable like the PVC. The connective pieces can be inset, which you will see in a little bit. And there's no need for a frame. It was just this plank of wood on these wooden blocks. The cons are that it needed to be treated. The wood needed to be treated in order to be waterproof. But that didn't take very long, and it was pretty fun to do. And it can be expensive depending on the price of wood at the time. We considered getting laser cut metal to get as precise as possible, but that turned out to be way, way too expensive. We got a quote that was about $600, and that does not fit with our affordability goal. So something else we made was a collimation slide. Also, you may have seen it on the back table over there. When you're working with lasers, it's important to collimate the laser, which basically means that the beams of light are all parallel to each other. So what you can do with this, you put the laser on the sled, you shine it at this target card, and when you move it back and forth across the track, if the laser point on the target card is of a constant diameter when you slide it back and forth, then the laser is properly collimated. And that's really important when you're working with lasers to have it collimated. So here's our focusing lens. Basically what it does is it takes this collimated beam of light and points it into a focal point at a fixed distance away from the lens. In this case, this is has a 25mm focal point. It allows for us to use a low-powered laser and still yield a high-powered response because the laser intensity is being focused at a single point. So it's really important when we're heating something that we have the target at that focal point, or at least near it. This is a plano-convex lens, meaning that this side is the plano side, it's flat, and then the convex side is like that. So this is our first 3D printed lens holder, also on the back table. It worked really well for our early tests. However, it was difficult to screw into the board. It was kind of bulky, so there was unnecessary filament use, which means that it cost more money and took more time to print. So what we wanted to do was make a slimmed down design, which is this right here. It works just like the older model. The inside is exactly the same. We added notches to make it screwing into the board a lot easier. And it was a lot more slim, so it took less filament, which makes it cheaper, and the print time was reduced by at least a few hours, which is really nice. Here it is from the top view, coming through the wood. And here is our DSU Bulldogs themed lens cap, which is important. Because like John was talking about, we can't see the light. So if for whatever reason it's on and we don't know about it, it's nice to have a lens cap to protect our eyes. Also, it keeps dirt and things from falling into the lens. So this was the commercial restrainer that we based our restrainer design on. They typically cost around $200, and this particular one, the one that we already had, was way too small for Rafu. So what we needed to do was we needed to make something cheaper. We needed to make something scalable, because if we make a 3D model of something, we can make it whatever size we want, depending on the size of our rodent. So this is our first 3D printed restrainer. It was still too small, and the print failed due to this large cavity here. So what we needed to do was increase the size, break it into smaller parts for easier printing, have thinner walls to save more filament, and add a front wall here to keep Rafu from walking out. So here we have a picture of Rafu. Here's the current restrainer that we have. As you can see, it's cut into 4 pieces to make the printing easier, and we just hot glued it together. We put some food in this tray. Rats are naturally curious, so once he was acclimated to the platform, he just walked right in there and sat there for like 10 minutes comfortably, which is really important. We want him to be comfortable. Also, I like how his nose pokes out of the front. It's kind of fun. This is another quick thing we made. So this is our proto board, which Carl's going to talk about a little bit later. We designed feet for the proto board because it's important that the bottom of the proto board does not touch any metal. This just shows something quick and simple that we can 3D print that makes our lives a little bit easier.
All right, so to power all the stuff that we want to use, we had to make a circuit that will power everything and do so reliably. So this diagram here on the right is a diagram of our final circuit that's powered by what's called an Arduino. It's basically a DIY-ist computer that you can program to do a bunch of simple things. Unfortunately, our project is simple. It's turning on a laser and turning it off just when we want it to. So what we did is we set up a circuit that has LEDs that change color so you can know if the laser's on, off, or in an in-between state. We have what's called a transistor. It's basically a different kind of on-off switch so that we can turn on and off the laser on demand. We have a photoresistor right here. That acts like a light sensor so that eventually when the laser trips it, when a rodent moves its tail, the computer can then know exactly when it was tripped, turn off the laser, and print how long it took for that reaction to take place. We also have a button because we still need a manual input to turn it on. And also this makes sure that we're safe, because if it goes on and off on its own, maybe if there's a glitch in the code, maybe you're doing something different for troubleshooting, it could go on and off and you won't know. So you need to be safe. Having that human control is always good. We utilize those 3D components, as Kobe pointed out, to protect the bottom of it, because if all those connections get joined together by sitting on a metal surface, it's not going to work properly. You can see here, this is our first design. It's literally just power, and as you hold the button down, it allows power to go to the laser. When it's not, it's off. Doesn't involve any code. And also we put it in our snack box. So when we buy from the company Thor Labs, they send us treats so that we want to go buy more of their stuff. And it kind of works. So we went from this design all the way to this final circuit. We can see that it connects the Arduino to the computer because the computer powers it as well as records data. And our photoresistor is on this long cable so that we can actually suspend it over the laser. Now, the circuit is also important because it makes sure that we reliably output a constant power to the laser. So if we didn't have this consistency. The laser could be on too high, too low, and then you don't know why the rodent's reacting. Maybe it's because of time, maybe it's because of power. So we made sure that we put resistors in the circuit, which will limit the power to ensure that it's always reliably putting out what we want. So we tested a couple different resistors. This line here on the left is a 40 ohm resistor, so that's higher than we wanted. So it made our power all the way down here, like below 20 milliwatts, which is really low. We then tried no resistance added, and we got up to about 120, which was too high for us. Then we kind of found our Goldilocks range. With a 10 ohm resistor, we got 60 milliwatts, which, as we zapped Dr. Krupka's arm, showed to be a really good setting for us to use. This is what we put the photoresistor on. It's a fairly simple design by Kobe, and it's fortunately able to break apart so you can store it more easily. And then you just run that photoresistor cable along and put it in this hole above the laser. So all of our pieces come together to make a really great design. You can actually see it in the back after our presentations if you like. So we have our restrainer, our platform, our little arch, laser, and circuit all coming together to be validated in the spring. So this is the protocol that we're going to validate in the fall. The first part we need to do is make sure that everything's set up for testing. This means getting the platform set up with the laser, making sure it's actually built. Maybe you had it taken apart. Then you, of course, have to put the rodent on to acclimate. Acclimation is really important with behavioral research because imagine if like one of us came in the middle of the night, pulled you from your house, put you somewhere completely different and started doing like testing on you, you'd probably react a lot different than if you were comfortable because the first scenario is really, really stressful. So by allowing acclimation time, we ensure that the data we get is accurate. We use a thermal imaging gun to take the temperature of the rodent's tail. This way, maybe if the basal heat temperature of the tail impacts how long it takes for them to react to the laser, we'll be able to know that and have that data recorded. Make sure that we take care of all the variables. And then we're going to mark the tail with India ink. It's basically hobbyist ink, but by adding this layer of dark ink on the tail, the laser is actually absorbed more superficially, but also makes a stronger pinprick sensation. So again, we can use a weaker laser to cause a stronger response. So that helps with the affordability aspect of our project. Then we have to upload and run some code. We have two codes that we have available right now. The first one is fairly simple. This is a snippet of it. Basically what it does is it turns on and off the laser for increasing intervals of time. So we'll turn on the laser for 1/10 of a second, have it shut off. And then the next time it'll go for 2/10 of a second. And we'll go all the way to 3 seconds until the rodent says, ow, that was a little uncomfortable. I'm going to move my tail out of the way. And then we have some data that we can use for analysis. It's about 88 lines of code, which may seem like a lot, but if anyone tuned into the math and computer science talk last week, they had upwards of 2,200 lines of code, which is a lot more than this. So fortunately, it's very simple, so it helps with the accessibility to maybe some labs that don't have some comp sci understanding. We have another code that uses our photoresistor. So instead of it just being discrete intervals of time, now it's waiting until the rodent moves its tail, is detected by the sensor, and then records how long it took for that to happen. So this is a little bit more precise, and of course, has a little bit more code and uses some more complex code using something called a switch case. And finally, at the end of the day, you always got to do some cleanup, perhaps the least favorite part, but it has to be done. So you're gonna return the rodent back to its little home, making sure that no damage was done. Of course, we've had all these safeguards as we've shown before, but it can never hurt to be too careful. Then you got to make sure you clean the platform. As we've alluded to, rodents do get a little messy, so you want to make sure that it's thoroughly clean. You need to make sure that all data recorded is in a permanent spot. The way that we currently have data printed is in a temporary way. It's like opening up a tab on your computer, then closing. Once you exit out, it's gone. So you need to make sure that's all stored properly. You need to properly store all your electronics, all your laser equipment, make sure no dirt or grime gets in there because you probably spent a decent amount of time getting it to work. And then finally, if you're done for a long period of time, fortunately, the way this thing is designed is that it can be easily broken down and stored away. So that's always another option too.
Okay, so I'm going to talk a little bit about some of the materials we used. As I was talking about extensively before, we made a lot of 3D design items. This allowed us to have more precision than just using hand power tools to try to build what we were going for. The other thing is these 3D files can be made available to the public, which helps with our accessibility. So we can just upload it to a database and people can download it and print it on their own printers. So we created a container to hold the collimation assembly focusing lens. Restrainer to comfortably house Rathew, the photoresistor scaffold, collimation apparatus, and the protoboard feed. The software we used was Fusion 360 by Autodesk to make the 3D design, and we used the monoprice flash print software to slice and print the components. The monoprice voxel printer would then take that file, print it out. Here's a picture of our printer. This is the filament reel. Here's the print bed. This is a cube trophy, and if you have questions about what cube is, we can talk about that at the end. Here's the view. This is what I see when I'm in Fusion 360 designing something. We had no experience with this program beforehand, so it was a pretty good learning experience. We had to make sure that especially this part that holds the lens and this part that holds the collimation apparatus was the exact dimensions that we needed based on the Thor Labs information that they sent us when we bought the product.
We also had a lot of different software. As Kobe pointed out, we use Fusion 360 and the monoprice software. But let's say maybe you have different preferences. Fortunately, there are a lot of other options. Fusion 360 is available for free. There are DIY versions and often available with institutions. Flash print is also free, but there are some other open source software. For example, Blender is an open source 3D design software. And because it's open source, there's often a lot of information available online. You can find a lot of people who talk about what they can do. You can find good tutorial videos, tons of information. Similarly with Cura, this is another open source. This is 3D printing software though, and it's often actually recommended by many companies because it gives you a lot more fine control over your prints, such as speed and how you want to support everything. We use the flash print largely because it has an easier user interface for people that were first getting into it. For coding, we use the Arduino Ide. This is just that little circuit board company's program for you to upload code. You could use Microsoft Visual Studio Code. Again, it just has a lot more options. So the Arduino Ide was a bit more simple for people first getting into it. And to design that pretty diagram we had earlier, we used this program, circuitdiagram.org. And as Kobe pointed out, we never had experience with any of these. So this, for a lot of us, was our first time seeing that intersection between computer science and biology. So it was a really great experience for us to see what else there is in bio and really help us to live out that DeSales mantra of be who we are and be that well as we continue to explore our interests in the field of bio. Now, we also had to buy a lot of physical components to make everything work. We had all the Thor Labs parts, so the laser itself, the lenses, the things to collimate it. We had the Arduino board. Again, this is also open source, so you can find a lot of hobbyist stuff out there. In fact, that's how we learned how to do some of our circuitry. And it's relatively easy to code, and even the boards themselves are really tanky. Like, you can hear stories of people spilling like tons of water and food on it, and they still work. So it's really good for a lab setting. We also had to get miscellaneous electronic components to wire everything up. Fortunately, they're often available in DIY kits. So all this stuff here is an image from Amazon of the kit that we bought for $22. It provides a lot of bang for your buck. So as a result, this summer we created a system to validate that is affordable, reliable, accessible, and safe. With affordability, we can take a look at some ways that we save some money. So here in the center is what's commercially available and what we could have bought. But on the right, this is what we did with some ingenuity and some time. So to hold the laser itself, we did acquire this to help us get everything set up. But instead of having a lab buy this for $171, we could 3D print something and go to the hardware store to buy nuts and bolts and do the same thing for $11. For safety glasses, we could buy ones that are $177, or we could get hobbyist ones for $40. Restraint tube, $200, or with some glue, some nuts, bolts, and some printing time, we can spend $16. A testing platform, maybe $600, or again, another trip to the hardware store, which we grew to love over the summer, $64. So in total, what we could have spent almost $1,200 on, we did for about $130. Now, we still had to buy a bunch of other components, regardless of the method we used. We had to buy the laser diode itself. We had to buy the Arduino. You can get other smaller boards, but the one that we got is far more versatile, and there won't be that much of a cost difference. Electronics costs about $50 if you don't already have wire. If you do have wire, that cost goes down. A thermal imaging gun, India ink, other 3D printed materials like that collimation assembly, and some specialized tools to help focus the laser and get it collimated. So in total, not including R&D. This is what we spent on our final product, $773. This is before shipping and tax and all that. But let's say someone's research doesn't need the thermal imager, maybe it doesn't fit within the scope of the paper, maybe there's research out there after we publish this. You could do this for $300, which is less than the cost of a new Xbox, which is pretty cool. Now, of course, this doesn't factor in things like buying a 3D printer or maybe some other tools that you have. But if you don't have something like a 3D printer, you can often find them available in certain places, or they're just really good investments to have. There are often 3D print file collections online for labs. So there's a lot of potential to use this in other ways. So it's a really good investment to have for a university.
And then just honing on the reliability and safety. So as Carl was talking about, we did use the Arduino, and that was the new computerized method that we were talking about for the automatic timing, as opposed to the manual timing with the stopwatch and the clock. Electronics have constant energy demand. So as he was saying about the current and using the resistance of the 10 ohms, we did create something that was reliable and that would be consistent. So if people set up their circuitry the same way and set it to the same current and with the same resistance, it will output the same power. The 3D printed materials were very precise. As Kobe mentioned, we did use 3D printing because that was way more precise than our previous models when we were trying to make things with PVC. Laser can work within the casing design. So as long as you have your focal point or your laser 25 millimeters away, it will output the same focal point. And then the wooden platform and restrainer can save for house rodents. These were more reliable than our previous methods as well. And for safety, just mentioning that we did use safety goggles for everything, and that the laser did create a pinprick sensation, but it was not harmful. In conclusion, we found that the IR laser was very effective for testing thermal nociception in a way that could test thermal nociception. It was affordable, accessible, reliable, and safe. And we devised an alternative to other less effective methods of testing thermal nociception, such as with the light bulb and then the hot plate test. Moving forward, in the fall, we would like to validate this and test on rodents, but this is just a tiny fraction of the bigger goal. We'd like to test thermal nociception to assess the efficacy of drugs for treating spinal cord injuries. Lastly, we'd just like to have some acknowledgements for the people that made it possible. Obviously, the women for DeSales, they funded our research. Without them, we wouldn't have this opportunity. Professor Karen Ruggles, who helped a lot with the 3D printing design. As Carl said, none of us had experience before coming here, so that was very helpful. Dr. David Starling, who helped a lot with the optics and with the lenses. Dr. Joshua Slee, the DeSales served faculty and staff, so Dr. Krupka, Dr. Beachboard, Dr. Huynh, Carol Ann, Ms. Maybrook, Mo Saad, and then the DeSales University administration, Brother Dan, Father Jim Greenfield, Miss Linda Zerby, and Mr. Michael Latham. And lastly, Matthew. Without him, he couldn't make the restrainer properly. He was just a good time. Are there any questions?
If anybody in Zoom land has any questions, you're welcome to put it in the chat. I have time for a few questions for this group. Our lovely advancement team of John, Dan, and Jackie are manning the information system here. For those that are in person, you can see the laptop set up. For those on Zoom, you can't. It looks like you know what we're doing here. Very professional. Any questions in-house or online before we move on to our next group? Dr. Mayville.
Guys, explain what the next step is and
Yeah, so a lot of drugs that you could use for treating spinal cord injury, like any drug research, of course, might have side effects. So when treating a spinal cord injury, one thing you might want to know is how much pain could this drug cause the user? So a good way to test if you're in more or less pain is how quickly you react to outside stimuli. So if you react, say, to our laser system at 0.3 of a second and you take the drug and now you react at 0.1 of a second, you're probably under more stress. And that way we can deduce that you're in pain, because we can't ask a rodent, are you in pain? We can observe how its behavior works. So, yeah.
So I think it's a really exciting project. I was talking to Dr. Krupka about it the other day, and there is plans to publish this work. So there is a journal where you can publish the development of these kind of resources and supplies. So we're hoping in the next semester these students will be published authors as a result of their work.
I do have one question from Diana. It says, did you consider the paints components?
Let's repeat that question. What was it?
It says, did you consider the paints components?
Oh, of like what's actually on the platform.
So just for the audience, everybody can hear it. The question from Zoom was, did you consider the components of the paint on the platform?
And added, it says, as you applied it to the substrate.
And as you apply it to the wood.
So we just got some plywood and we used decking varnish that was rated for outdoors because that would like weatherproof it. So we figured that would help get like a waterproof base down. Then we used what's called Plasti Dip. It's a spray on rubber. So we could have like a latex covering that would help everything be safer. So we opted to make sure that we could waterproof as much as possible, because if not, urine could soak into the wood, which would be bad.
Great.
Okay, I think we'll move on to our next group. Let's give them one last round. All right, our next faculty research group is Dr. Dia Beachboard. She is an assistant professor of biology. She is our microbiologist and virologist, and she happens to study the coronavirus. We're very fortunate to be able to hire her just prior to the pandemic with her expertise in coronavirus, which has become the topic of everybody's dinner tables. So I'll bring up Dr. Beachboard, and she will introduce her students.
Okay, so like Dr. Slee just said, I am a new faculty, so I just started last fall, and this summer research was actually really helpful because not only did the students get to do research in the lab, but they also got to help me set up the lab. So I didn't have to do that by myself. I had these wonderful students that can help me with that. And so I have 3 students. Their names are up here. You guys can make your way up here. They are Jared Franges, Brennan Garancher, and Dee Kumar, and they're going to talk about this project that we have started, looking at how coronaviruses evade innate immunity using their protease to cleave host proteins. So it's really important that whenever you get infected with these viruses, that your immune response is able to kind of shut down that virus. So we're looking at how the virus is actually counteracting that response. So I'm going to turn it over to them. Get out of the way.
Hi, I'm Jared Franges. I'm Brennan Garancher. So just as Dr. Beachboard said, our project is on coronavirus evasion of antiviral innate immunity through the cleavage of the host proteins, and we'll get started.
So humans and viruses have an ongoing battle, and humans have evolved in many different ways to fight off viral infections through innate and adaptive immunity. But viruses have been evolving along with us and have ways to fight off our barriers of infection. So these barriers of infection include intrusive immunity, which is always present, and that includes physical barriers like your skin or mucous membranes that trap pathogens. Innate immunity happens at the cellular level. This includes different signaling pathways that will prepare the cells to fight off infection. And then there's adaptive immunity, which is where your immune system remembers a previous infection and is now able to more efficiently fight off that infection if it happens again.
All right, so going back to that last slide, this is just going to be another iteration of that last slide. In this case, this is more of a timeline iteration. So it goes from infection all the way down to your infection outcome. So within minutes to hours, your intracellular innate immunity will activate. And that intracellular innate immunity will activate the killer immune cell response as well as your adaptive immune response. If you ever learned about T cells and B cells, that's the adaptive immune response. And then all of these things will lead to your infection outcome, whether your host cells beat the virus or if the virus eventually beats the host cell. So within minutes to hours, this intracellular innate immunity activates. And that is very important because if your host cells do not recognize this virus, all of these downstream pathways will not be activated and you won't be able to fight the virus off. So how do these cells sense these viruses? They use these things called pattern recognition receptors, which we will refer to them as PRRs the rest of the presentation. So these are in your host proteins. These are host proteins in your host cells, and they will bind to viral components and transmit this downstream signaling. And these viral components we will call pathogen-associated molecular patterns, PAMPs. And these are common features of viruses, such as DNA and single-stranded as well as double-stranded RNA.
So as Jared said before, we have PRRs to recognize all these different PAMPs and viruses. So this diagram just basically shows how or where each of these PRRs are located in the cell. So the blue ones will recognize the RNA PAMP and the green ones will recognize the DNA PAMP. So the virus enters the cell in through the endosome. And the endosome has these receptors for both RNA and DNA. And then the virus will then come into the endosome and then enter into the cytoplasm where both RNA and DNA receptors are found. The RNA viruses tend to replicate in the cytoplasm, and the DNA viruses tend to replicate into the nucleus, which is why there's only a DNA receptor, DNA receptor in the nucleus. Sorry, the remote stopped working for a second. So this diagram now shows just basically where the viruses replicate and how they enter again. So again, they enter into the endosome. Both RNA and DNA viruses do come in through the endosome. Then they will enter into the cytoplasm of the cell, where I said before, RNA viruses will tend to replicate in the cytoplasm. Then the DNA viruses will then tend to replicate in the nucleus, with the exception of pox viruses, which are DNA viruses that replicate in the cytoplasm, and the influenza virus, which then replicates it's an RNA virus that replicates in the nucleus.
So the innate signaling pathway, this is what it looks like. So this is a very important part of your innate immune system. So this is a receptor. This is what's going to detect a virus that comes into the cell. But there's the adapter that brings along the rest of the signaling pathway to a common location. And the rest of the signaling pathway includes like different kinases. These ones are unnamed for now because there can be multiple. And those induce a certain reaction from the cell. And for this example specifically, it's moving a transcription factor into the nucleus to induce the production of interferon beta. And interferon beta is a signaling molecule that will tell other cells to go into an antiviral state, which basically means it's like a messenger from a first little village which is your host cell, going to surrounding villages, surrounding cells, and telling them to get their armory ready to fight off some viral invasion. So Rig-I and MTA5, which is not shown, are the PRRs that Jared and Brennan have been mentioning. And they'll detect the virus through their single-stranded or double-stranded RNA. And once those are activated by that RNA, it'll send a signal down to MAVS, which is our adapter. And that will call on the rest of the signaling pathway. For this one specifically is NEMO and TBK1, which are just different kinases that will induce the IRF3, which is our transcription factor, to move into the nucleus and induce the production of interferon beta, which is, like I said before, the signaling molecule for the rest of the the rest of the cells. But viruses, like I mentioned before, also have also adapted to fight off our barriers to infection. So poliovirus, HCV, and HIV-1 all inhibit the pathway in different ways. So HCV has proteases that will cleave the adapter at certain sites, and cleaving that adapter will make it non-functional, making the pathway incomplete. So the transcription factor won't make it to the nucleus, and you can't have the production of interferon beta. Same thing with poliovirus and HIV. They don't specifically cleave those proteins, but they do inhibit them in certain ways, making them non-functional. It's also important to note that once a protein is cleaved or made non-functional in the pathway, anything downstream of it won't be functional or won't be activated anymore. So it's an incomplete pathway and you won't have the production of interferon beta. So for example, if HCV cleaves the adapter, then anything downstream of it won't be activated, you won't have the production. But anything above it, so like the receptor, it'll still be functional and active, but it's not going to.
So in our lab, we work with coronaviruses specifically. So coronaviruses are a positive strand RNA virus, which means that there is no DNA involved in it whatsoever. And positive strand means that they can just start making proteins right as it enters the cell. So they're a virus that has about 27 to 32 kilobase genomes, which are some of the largest genomes for viruses in the world. And they infect a wide range of animal hosts, including mice, whales, cheetahs, deer, birds, and humans. And right now, there are 7 known human coronaviruses. And these first 4, they all cause symptoms pertaining to the common cold. And if you've seen the meme before the pandemic that Lysol lists on the bottle that it fights human coronaviruses, it was targeted for those and not towards specifically COVID-19. And so we have 3 more human coronaviruses, SARS-CoV, MERS-CoV, and SARS-CoV-2, which in today's world is known as COVID-19. And those cause severe lower respiratory infections, which can actually be fatal to humans. So in our lab, we work with murine hepatitis virus, which is a virus specifically, it infects mice. So it's actually really safe to work with in our lab. So if it would happen to get into our bodies, it wouldn't actually do anything because it only has the receptors to target mice. And it's used as a base model for coronavirus replication in many laboratories.
All right, so just as Brendan just said, we're using murine hepatitis virus, very safe for us to use. So this is the genome of this virus. So off to the right here, those are the genes that encode for structural and accessory proteins. So if you see on the news, the little ball with the spikes, that gene right there encodes for the spike protein that you see. And then off to the left here is what we're really interested in. These are the genes that encode for non-structural proteins. And this will actually be translated out into one large polyprotein, such as this. So it will translate out into one single protein, and then within itself, it has proteases. So non-structural protein 5, or NSP5, right here, highlighted in blue, is the main protease. And this protease will actually cleave its own protein at 11 sites marked by these blue triangles. And then it will also, there's another protease over here that cleaves the rest of it. So then these proteins will go off and have their own functions, but the one we're really interested in is the one highlighted in blue, NSP5. And NSP5 can cleave your host proteins as well in that innate immune pathway that Ide discussed. That NSP5 can cleave those and inactivate the innate immune pathway. So what they did is we have a consensus site that is made off of these 11 cleavage sites on this specific coronavirus, and also including all other coronaviruses which have the same sites, just slightly modified. So this is the consensus site. So as you can see, it's pretty general. So off to the right here, we have some areas where it can be really any amino acid. Off to the left, we have pretty specific amino acids here. We need leucine and glutamine off to the left here. But this one could be any amino acid. This one, a range of 3 amino acids. So we have a pretty good idea of where NSP5 will cleave, and this can cleave on the virus's protein, the polyprotein that I showed you, and also the genes of interest that we're going to talk about, which is coming up next. So we can actually predict where NSP5 will cleave these proteins using some bioinformatics. So the first step of this process is using a system called UniProt. And this is how we can look up the amino acid sequences of the mouse and the human cells. So we're going to look up the amino acid sequences of the innate immune proteins that Ide discussed earlier. Then we can align them between the mouse and the human genes. We can align them using +L-W to see how similar they are, see which proteins are conserved between mouse and human, and see which ones might differ. So then we can put these amino acid sequences into a server called NetCorona, and NetCorona will do all the work for us and give us a nice output of predicted cleavage sites for this NSP5 protease. So we can put in all of our host proteins, see where NetCorona thinks it'll cleave that protein, and then we can go from there. So an example output of this would look like this. So up to the left here is the amino acid sequence that you input. So it'll spit it back at you so you can see it. And then the bottom graph here shows the sites on that amino acid sequence that it will cleave on. So it needs to be above a threshold of 0.5, which is a probability score. And it will give you the position and the sequence that NSP5 will cleave the protein, or is predicted to cleave the protein, if the threshold is above 0.5. So here on this example, there are 2 sites on this amino acid sequence that NSP5 will cleave. And they're off to the right there. You can see the specific position and the exact spot that NSP5 is predicted to cleave these proteins. So these proteins we can put into this net corona. So we did them for a lot of proteins. And we got these 5 proteins that NSP5 is predicted to cleave on the mouse. And then we repeated this process for the human equivalents of these proteins. So as you can see, some differ, some are the same. So with MABs, you can see the mouse has 2 predicted sites, but the human has 0. And with the alignment, we also saw that MABs was different between human and mouse, but TBK1 is highly conserved. It has 1 cleavage site, predicted cleavage site for mouse and human, and that cleavage site is exactly the same. And with the +all W alignment, we saw that they were very similar with TBK1. The same is true for IKK epsilon. Nemo is not highly conserved. As you can see, we have 2 different sites for the mouse, but 0 for the human. And then STAT1, we have the same cleavage site again, and the alignment also showed that they were very similar. So why is this important? It's really important because coronaviruses can emerge out of the animal versions and jump to humans. Now, we fully do not expect the mouse version that we're working with to jump to us and infect us. We fully do not expect that, but it does happen. So it is important to note that we can study how NSP5 cleaves these proteins and inactivates the pathway, and it can be applied to see how these coronaviruses can also inactivate our innate immune response.
Now we'll talk about cloning. And so the mouse genes that Jared just mentioned, we clone them all individually so that we can put them into certain cells and test their functions in those cells and like where they're cleaved and all that. So the first thing that we want to do is that we get a plasmid that's pre-existing that has this flag tag that we'll be using later to do a Western blot, which Jared's going to go over. And then it has like this excess gene area that we're going to cut out. So that's what we'll do first. So to cut out that gene, we have NOT1 and TME1. Those are our enzymes. And they'll cut at these specific restriction sites at the beginning and the end. And this plasmid is specifically designed to have those restriction sites to cut out that gene really nicely. To show that, like physically, we have it on this gel. So this back piece right here is called the backbone, and that's right here. It's the top bigger band, and we harvest that from the gel by cutting it out of the gel, and we'll use that for later. And then cut out this gene, which is not useful. And then we have our gene of interest that we're going to paste in there, and it has these 2 little side pieces right here that are just extra sequences that help it attach to the backbone. And the gene of interest right here is MABs, but like Jared mentioned, there's like 5 others as well. And there's also viral RNA ones that we have and the mouse ones like MABs. And so to do that, we have to amplify them first. And that's shown on these bands right here. Some of them are a little bit faint, but it happens. And we have these 2 right here, which are pretty good. So good enough. So the next step that we did was take a gene of interest that we had and the backbones that we had and paste them together in a process called ligation. And to do that, now we have like a full plasmid that we can put into bacterial cells and amplify them again in the process called transformation. And so this is a summary chart of all that we've been doing with those cells and the plasmids. So these are the 3 viral RNA that I was mentioning, and then these ones are the mouse ones. So the first thing that we did was a PCR extraction, which is amplifying the backbone and the plasmid, and then getting them on the gels, getting them out, cutting out the pieces that we need. And then the next step is ligation, which is pasting the backbone and the gene of interest together. And then lastly, it's transformations, which is putting those plasmids into bacterial cells and then having the bacterial cells amplify those up, and so that we can use them. So that's going to be the last step, which is pulling the plasmids out of the bacterial cells and then putting them in other genes so the proteins can be expressed in that, so we can see if they're cleaved right, if we have the right insert, and to test other things.
So we do have some future project directions that we want to take into effect in the fall. So we want to show how NSP5 cleaves different proteins on the innate immune response pathway. So we would use those genes of interest that we did through the cloning process, and then eventually introduce them to the viral protease NSP5 to see basically where it would cleave on that plasmid, and then we would identify that on a Western blot that we would run eventually. But we can also mutate the protease NSP5 to where it cannot cleave any of the target proteins, and we can also mutate the target protein to where the protease NSP5 cannot cleave it either. So as I said before, we were going to run a Western blot. So this is an anticipated example of what we would see if we were to run a Western blot on all these gene products. So we would have it identify any of the different gene products that we studied, and then we would either introduce them NSP5 up here, or we would leave it without NSP5. So if we leave it without NSP5, then we wouldn't expect it to cleave anywhere. So we would see these bands much higher than any of the other lighter bands because it would be still a full-size plasmid. So we also introduce it to an antibody that binds to flag so that it can be expressed on this Western blot. So as we see here, all these darker bands all have the full-size plasmid. They have not been cleaved. They also have not been introduced to NSP5 either. And these lighter bands show that when we introduce these different gene products to NSP5, we would see a lower band because the plasmid is smaller that has the flag tag attached to it. So in the case of TBK1, you see that there's no band in the NSP5 plus lane. That's because it cuts around the second to third amino acid, and that would be way too small for us to actually visualize on the Western blot, so it wouldn't show up at all. Now this lane down here shows that we actually introduced NSP5 into that different lane. So you'll see that the plus lanes all have the NSP5 band, while the minus lanes do not have a band. The tubulin lane down below shows that we added an equal amount of sample to each of the wells so that we can get the most accurate results. So we also want to show how NSP5 inhibits IRF3 activation. So we also grew up some human cells and then introduced them to GFP IRF3. GFP standing for green fluorescent protein, so we're able to visualize them under a fluorescent microscope. And IRF3 is that transcription factor that will eventually move into the nucleus and promote interferon beta production to warn all the other cells that it has been infected with virus. So this first image is what you saw on the previous slide. There is no virus being introduced to the cell, so there's no need for the GFP IRF3 to move to the nucleus to start making interferon beta. So all the GFP IR3 will still remain in the cytoplasm, making under a fluorescent microscope, making the cytoplasm of the cell glow green while the nucleus remains dark. In this one, we introduce a virus that does not inhibit the pathway at all. So the GFP will be able to freely move into the nucleus because it has detected a virus and the pathway has started. And then it will promote the interferon beta production, allowing the nucleus to glow green because all the GFP IR3 is in the nucleus while the cytoplasm remains dark. And in here, we introduce a virus that does actually inhibit part of the pathway. In this case, it's the adapter protein. So the GFP IR3 will not be able to move into the nucleus because anything downstream of that adapter will not be able to be activated. So the GFP IR3 will remain in the cytoplasm, making that glow green while the nucleus remains dark.
All right, so just a quick summary of what we've done this summer, what we're trying to look at. So as we said in the beginning, your host cells are in a constant battle with the virus infection. So the innate immunity pathway is super important in being activated, activating adaptive immunity, activating killer immune cell response, overall just fighting that virus off. So that is why we are studying this innate immunity pathway and how these viruses can actually cleave proteins on that pathway and ultimately inhibit that pathway. Specifically, we're looking at that NSP5 protein I talked about earlier. We're using bioinformatics to predict these cleavage sites, see where it can cut, compare between human and mouse proteins, so we can apply that to maybe some human coronaviruses. And then physically, we need to actually clone these genes out like Needy described. And so we can use them to be tested, and how Brennan explained, we're going to be using a Western blot to see if NSP5 can cleave those proteins, and we can see the IR3 activation, the interferon beta induction, all of that, so we can ultimately see how these viruses can inhibit that innate immunity pathway.
We'd like to acknowledge everyone that made this program possible, especially the women for DeSales for funding our program, and then DeSales University for supporting us throughout all of this whole process and helping us through everything that we needed.
We have time for a few questions. If we have any from the in-house audience or our Zoom audience, Dr. Barnett.
Yeah, could you go back to the genome screen? So the open reading frame is the second one. Is that does that start at a different reading frame, or am I?
Yeah, I believe the overlap there actually indicates that it does start in a separate reading frame. It's not perfectly linear. It does actually start in a separate reading frame.
Steve Opreska here. What commercial item do you have available to do this gene splitting?
I think everybody knows what a minus one factor is. Just so simply done. Any other questions from in-house or online? Speaking of coronavirus, it's a good kind of plug for one of our previous virtual scholar series that Dr. Beachboard and I did, where we talked about the coronavirus and the vaccine. So I know Advancement still has that up on the website. If you missed it, it's still there. You're welcome to take a look at that. Otherwise, let's thank our group here again. All right, so I hope you have all started to appreciate the amount of work that goes into the research labs here at the Sciences at DeSales. To remind you, these students have been working for 8 weeks to get this work done. So the impact that it has on the faculty members' labs is just profound. All of the faculty that participate are junior faculty here. Some of them, this is their first year here, going into their second year. And just the ability to have 3 students help them set up their labs has been very impactful. It's really advanced their professional development significantly. Last but not least, we have another assistant professor of biology, Dr. Alex Krupka, who will talk to you about his chemical ecology work.
All right, thank you, Dr. Slade. All right, so we're the last group up. My name is Dr. Huynh. Again, came in the same year as Dr. Beachboard, so we're the newest faculty in our department. So this was a really great opportunity, not just for our students, but for us as well. Again, just echoing what's been said. To get a lot of material, set up our labs, get our projects going. I think that my group has done a lot this summer. These 3 students were in my research lab for the spring. They will also be returning for the fall, so we'll be continuing a lot of this work. They've been doing a bunch of different things this summer, so you might have seen them hiking around campus. We've been over here in Herd doing some molecular work. We've been on the 2nd floor doing some chemical work. And we've also been over in Campbell doing a lot of computer coding and statistics. I won't go into the details. I'll let them do that. My 3 students are, I have a rising sophomore, Marcilla, and 2 rising juniors, Katie and Caitlin. So I will turn it over to them and let them explain all the details of what we mean by this field of chemical ecology.
Yeah.
Stop the share and then do it again. Just make sure you have computer sound checked.
Yeah.
Video.
Yeah.
So stop share is.
You have to move it. Do it now.
bear with us as we reconfigure the mainframe here. Even though
No, I know what you're trying to do. So we can't play the media. I changed it. I changed it. Yeah. Just request access. Someone's gonna have to log in on their phone and get access. Yeah. So we can do that. Log out and log in as yourself.
Yeah, we could do that too. We could just log in as yourself. Yeah, just sign on.
So while they're doing their thing, the video is up and running. Those people that study animal behavior always have fun videos. Videos kind of. So I just wanted to thank, there are some other important people in attendance today. So this was kind of our first informal meeting of our science advisory panel. For the biology and chemistry, so there's a couple members in-house and virtually. So thank you for coming, and I hope you are impressed with what you see. More information coming your way. So, the other thing I wanted to acknowledge is the funding sources for this really are the university and for DeSales, but also we provide funding on the endowments that we have in the sciences here. So we have the Dr. Roger Bird endowment for undergraduate education, and we have the Dr. Harry Warren endowment for natural science equipment. We do rely heavily on both of those endowments to support research and the undergraduate experience here at DeSales. Those endowments are so strong because of the friends and alumni of DeSales that contribute to them. So we really do appreciate everything that's been done for those endowments. And it looks like we're back up, so I will turn it over to them.
All right, so this is our presentation on avian chemical ecology. Like Dr. H said, I'm Katie.
I'm Caitlin. I'm Marcella and Wynn.
All right. So what is chemical ecology? Our field is focusing on chemical ecology this summer, which is just how chemicals will affect the interactions of different animals. And an easy example of chemical ecology is a picture here where ants will follow one another based on their trail pheromones, which is studied by scientists to understand how that process works better. Specifically though, we are focused on the chemical ecology of birds and how birds use their sense of smell to detect chemical signals. Normally when you think of birds, you'll think of their really bright feathers and the sounds that they make. But we specifically are focusing on how they will use their use of odor. And it has been, more information now has been gathered on the use of their odor systems to drive their behavior in stuff such as mate choice, kin selection or kin recognition, aggression, foraging, all that stuff. I'm not. Oh, there it is. So there was this paper that was published just about 2 weeks ago in the journal Nature. How birds can use and manipulate their smell in this cool way. So these European white storks have actually been found to cue in on the smell of freshly cut grass. And they have been found to associate this odor with the disturbed population of insects and rodents that burrow deep in the tall grasses of fields. And when the grass is cut, the insects and rodents are now exposed, so they can associate this smell of freshly cut grass with a good population of prey for them. So throughout the course of the summer, we did 3 projects focusing on chemical ecology. So the first project looked at herbivore-induced plant volatiles to see if birds can use their odor cues to associate with prey. And the second project we did was a genetic study where we looked at the genetic makeup of the local chickadee population on campus. And the third and final project we worked on this summer was just the preen oil chemistry of birds
specifically looking at their chemical composition. And these 3 experiments, while they are very different in scope, can be used together to eventually better explain the chemical ecology of songbirds, both here and other places.
All right, so the first project that we completed this summer had to do with herbivore-induced plant volatiles. And because that's a bit of a mouthful, I'll be referring to it as HIPVs from now on. HIPVs are basically these chemical compounds that plants release upon wounding, whether that be by an insect or some kind of other herbivore. And they can function in plant communication as well as act as hormones that can stimulate biological pathways that produce defensive compounds. It can also act as a sort of indirect defense. As you can see with the parasitic wasp there, it can have effects on herbivore physiology and recruiting of predators or parasites. So basically what these parasitic wasps do is that they are attracted to these certain compounds that plants release when these aphids come and feed on them. These parasitic wasps then come and plant their eggs directly into the aphids and basically kill them as the eggs grow inside the aphids. So it's pretty scary stuff, but it's also kind of cool. So besides studying how cool these HIPVs are, we want to know specifically whether or not birds can cue in on these HIPVs, specifically whether they can use them to locate insect prey. So there are a variety of HIPVs, but we're focused on these 2 specific HIPVs called methyl salicylate and methyl jasmonate. These are very general chemical compounds that most plants produce. And just like other HIPVs, they're known to act in recruiting predators and parasitoids, as well as being released when plants are wounded. So again, these 2 specific compounds are the ones that we're focused on. We want to see whether or not birds can cue in on these 2 HIPVs. So here are a couple of pictures to show how we set up our experiment. This picture here just shows our clay caterpillars that we set up. They're just a couple centimeters long, and they're in this little inchworm shapes to fulfill our crafts and arts and crafts directive. These were made out of clay specifically so that we could see visual evidence of predation on our caterpillars. If we use plastic, for example, we would not be able to see peck marks or bite marks on the caterpillars, so we would not be able to quantify it. This picture right here shows the bottles that we use that hold the HIPVs, whether that be methyl salicylate, methyl jasmonate, or hexane as our control. We placed capillary tubes in them in order to control a slow release of these HIPVs. We didn't want them to disperse all at once. We wanted them to sort of disperse aromatically over a course of a couple days, because we did conduct this experiment for a period of about 2 weeks. And as you can see, we used parafilm to attach them onto the trees or whatever plants that we use. Here's just a picture of our caterpillar next to a cicada. The cicadas were our obnoxious companions during our expeditions outside to each of our sites this summer. And you can see they're about the size of the cicadas, so they're pretty small. Here's just a map of DeSales. I believe we are right here in the Herd Auditorium at the moment. We chose a variety of locations for our project. So each of these stars represents a specific location using one of 4 host plants that we chose this summer, either red maple trees, black walnut trees, wheat, or cattails. And mainly this was chosen due to ease of accessibility as well as abundance across campus. So for example, this little spot right here, I can just stop shaking, is the site by the pond that contains our wheat location. And this one right here is the black walnut location. So to explain this a little further, because I know it's a little bit confusing, we're going to go ahead and look at this site behind the Aviat parking lot and show you exactly what's going on at each location. So as you can see here, this is one of our red maple locations, and we chose 3 trees separated about 20 meters apart. Each of them contains a different bottle of treatment, a different bottle of HIPV. This one contains methyl jasmonate, this one methyl salicylate, and this one hexane as the control. Each of the trees had 3 caterpillars on them. That's basically how we set up our experiment for each of the sites around campus. So again, the Aviat site was just one of our 15 locations that we chose across campus. All the other locations had one of 4 of these 4 different host plants, and I just have a couple of pictures to show how we set them up. You can see the little caterpillars with the inchworm shapes on the branches, on the red maple, and then on the cattails, the wheat, and the black walnut trees. So we collected data every other day for about a period of 2 weeks, focusing on 3 different questions and 3 different quantities. So we wanted to ask how many caterpillars were pecked at each site, how many pecks in total were found at each site, and whether or not the site was visited, which we quantified later as the total number of visits to each site. So we got a lot of good results actually, and we got a lot of pretty cool pictures. So our poor caterpillars were pecked, as you can see in this picture, in this picture. Some of them were absolutely mutilated. And some of them looked strangely like they were just sliced cleanly in half. That was a strange phenomenon, but we'll go into that later. So just to begin speaking about the results that we gathered from our data, here is a graph showing the caterpillars pecked versus the host plant on the x-axis. Of course, with the black walnut, cattail, red maple trees, and the wheat. The 4 different colors, or sorry, the 3 different colors show the 3 different treatments. And for this particular one, I believe that there was a significant effect of the number of caterpillars pecked, or the host plant on the number of caterpillars pecked. So as you can see, there are some trends here with the black walnut trees, the caterpillars pecked being much higher than that of the wheat, which was very interesting. There was also a nearly significant effect, I believe, of the methyl salicylate against the control. For the total pecks versus the host plant, fortunately, we did not find anything statistically significant. However, there was a near statistically significant difference between the total pecks for the methyl salicylate in the black walnut versus the total pecks of the control at the black walnut locations. And again, for this one, you can see for total visits versus the host plant, again, it seems like the tree species have a lot more total visits than the herbaceous species. So I know statistics is kind of boring, so if you didn't really want to read the graphs, that's fine. I'll explain to you that the most important points to take away from our data is that the host plant has a significant or near significant effect on predation. And the significant differences between the predation in the wheat and black walnut locations are particularly interesting because they were very different, with the black walnut locations having a lot more caterpillars pecked, a lot more pecks, and a lot more visits than the wheat locations. The same thing can be said with the treatment, the effect of the treatment on predation, although this was merely a nearly significant effect. There was a big difference between the methyl salicylate treatments and the control treatments. So of course, this was a really cool experiment to carry out, especially because we got to go outside a lot and put up the caterpillars ourselves and see the pecks on the caterpillars. But we would like to extend our research in the future, and we think we could improve upon our research by maybe studying exactly what HIPVs birds use best to locate insect prey. For example, there are some HIPVs that are more specific to plants. We use simply methyl jasmonate and methyl salicylate, which are very basic HIPVs that are found in most plant species. So in the future, we could try using ones specific to corn, for example, and see how that could affect farming techniques as well. And in the future, moving forward, we'll probably focus on tree species, as the trees seem to give us more clear results and were more effective as host plants. So in the future, we might use, for example, just black walnut trees instead of the 4 host plants that we chose.
Okay, so the next question we asked was about chickadee genotyping.
And we asked, what does the chickadee population around DeSales look like, and how can this knowledge aid in future studies of chemical ecology? So you may be asking, why chickadees? Well, there's two types of chickadees in this area. There's the Carolina and the black-capped. The Carolina is from the south and the black-capped is from the north. And in the area where these two species meet, they hybridize, meaning they mate and have hybrid babies. And it has been shown to be moving north, this hybridization zone. So we want to know where the DeSales campus is in relation to the hybrid zone. And this relates to chemical ecology because chickadees have been seen to use chemical odors in their everyday lives throughout their entire lives. So it's a good species to study for chemical ecology. So the first method is we have to catch the chickadees, and I have this little silent film. So if you could not tell, that video was reversed. Unfortunately, we are not Disney princesses yet. So to actually catch the chickadees, we painted clay models to look like them to attract them, and we also play an MP3. Now we'll see if the audio works. To show you what the chickadees actually sound like. Very quiet. It is quite quiet, but nevertheless, we play it very loudly in the forest so the chickadees can actually hear us. And then we wait for a response from the chickadees. And once we hear it, we set up a mist net, which is like a volleyball net that runs all the way to the ground. And we basically sit in the forest and wait for chickadees to fly in and look at the fake chickadee. Now, these are the 3 chickadees we caught over the last 8 weeks. We also caught chickadees throughout the semester, so we had 2 more chickadee samples that we used in our experiments. Now, to genotype the chickadee, we took a blood sample from each chickadee we caught, and we extracted the DNA and then amplified the DNA using PCR. Then we cut the DNA based on its sequence and then ran the DNA through a gel to see the DNA fragments. Now, to cut the DNA, we focused on the SNPs, which are the single nucleotide polymorphisms, which are the point in DNA where one base pair is different. And then we take a restriction enzyme, which detects that specific sequence, and it cuts it in half. Now, we can use SNPs because they differ between species. So we know that, for example, the black-capped has a T while the Carolina has a G. So the black-capped with a T, the restriction enzyme will see that T and know to cut it. And as you can see in the diagram, at the bottom, the black-capped will have DNA that's lower on the gel because it's been cut and it's smaller, and the Carolina will stay at our control. So when we ran the gels, this was all the data we got. We used 8 different locations, each with its own restriction enzyme. And 7 out of 8 of them worked. So we used the 7 for our final calculations. But this image is very busy and it's a lot of data, so we can just zoom in on the green. So if you don't know how to read a gel, each column is its own chickadee. And each column has the DNA. So you can see in the green squares, that's each a piece of DNA. The Carolinas were the uncut in this example. So you can see on the left that there's 2 uncut, and then the black-capped is in the middle. You can see it's lower down. And this image is actually pretty interesting because we have examples of cut and uncut, but also hybrid chickadees, because they have both cut DNA at the top and uncut at the bottom. So if you don't care about gels and they mean nothing to you, that's okay, because we made a summary table of all of our results. You can see that each chickadee, we wrote down whether it was a Carolina, a hybrid, or a black cap. Sometimes it didn't work, which is why we have the NA. And then you can see at the bottom that there's a row of all the percentages of how much black cap DNA it has. So if it's the closer it is to 100%, the more it is black cap, and then the closer to 0, it's Carolina. And you can see in the middle that chickadee 3 was our only 100% purebred species. It was a black capped, and the rest were hybrids. Now, it's really interesting because the hybrids aren't 50-50. They aren't 50% Carolina, 50% black capped. They're actually closer to having Carolina DNA, which helped us to conclude that the hybrid zone is in fact located over the DeSales campus. And we can see that we are more south of the hybrid zone because the hybrid chickadees are now backcrossing with, the hybrids are backcrossing with Carolinas and creating more Carolina DNA-based hybrids. So for further research, we would know to go north to catch black caps and south to catch Carolinas, but if we want to work with hybrids, we know that they're right here on campus.
So the last project we did in the summer looked at the preen oil chemistry of songbirds, and preen oil is produced by the preen gland, which is right here. And it's located on the back side of the tail in almost all birds. And the preen gland will secrete the preen oil, which is just a thick, waxy oil. And in the act of preening, the bird will stick its feather or stick its beak over the preen gland, gather the preen oil, and spread it all throughout its feathers. And historically, this has been known to be important in waterproofing and cleaning their feathers, but through recent research, it has been found that these preen oils contain chemical components that have been really important in communication. And it's also important to know that the composition of preen oil varies between species of bird, while also varying between the sex of the bird and throughout different seasons that the sample's collected in. So our group is eventually interested in being able to answer all 3 of these questions, but these questions are really big picture and will require many consecutive years of research before we will be able to answer them definitely. So although we didn't get answers, we spent this portion of our summer just piloting methods to get us closer to eventually answering these questions. Like Caitlin mentioned before, we catch birds. We spent a good amount of time catching chickadees, but we also caught blue jays, catbirds, and this is a titmouse up here. And to catch these other bird species, we, instead of using an MP3 and a model bird, we just put the mist net up by a bird feeder and lure them in using a food trap instead. So we decided to pilot preen oil samples on 3 different bird species. We have a red-winged blackbird, a female house finch, and a male house finch. The way that we analyze chemical composition is through a method of gas chromatography mass spectrometry, which is abbreviated GCMS, which is broken up into 2 different steps. We begin with an oil sample, which we collect in the field from the preen gland. There's not much oil in a sample, probably about like a microliter in size. So we dissolve it in a solvent, and that is, we use dichloromethane, but you can use a couple other organic solvents. From there, you run the GCMS on the sample. The first collection of data yields a gas chromatogram, which is this image right here, with each peak corresponding to a different compound. In this example, there are compounds A, B, and C. So this hypothetical oil sample has 3 different chemicals that make up the oil. Each different compound is then run through a mass spectrometer, yielding different graphs for each different compound. So in this sample, you will run your oil and get 3 different peaks on one GC graph, and then generate 3 different mass spectra from your sample. And together, this information will allow us to identify the compounds that compose the preen oil, which is important for moving forward.
So now we're gonna look at the results of the male and female house finches. These are just two of the individuals that we piloted methods on. Again, these are gas chromatograms with each peak corresponding to a different compound within the sample as a whole. The baseline here, this low horizontal line near the x-axis, looks really fuzzy, which means that the oil sample was not concentrated enough, so the extraction methods probably need to be altered. Additionally, you can see that the female house finch ends its run in the middle of a peak, which just means that we probably didn't run the gas chromatograph portion long enough, and we need to run it longer to potentially be able to identify more compounds. And overall, just the noisiness of the baseline right here and right here indicate that the methods specifically for our house finch species need to just be altered a little bit. Now this is the image for the gas chromatogram of the red-winged blackbird. Again, each peak on the gas chromatogram is a different compound in the preen oil sample of it. And as you can see, the baseline of this gas chromatogram looks a lot better than it of the house finch, which is interesting because we ran the same methods on both. It can be easily counted from this graph that there are 13 different peaks, which correspond to 13 different compounds within the sample. And overall, this method seemed to work really well for this species, but it didn't work for the red-winged or the house finches, which mean that this method that we run might need to be tailored to be species specific. So based on our results, it is known that we need to collect more samples for a larger sample size. And we also need to create a more diverse sample. So collect them at different times of the year, different sexes of each bird. We also need to establish the best extraction method for GCMS. Some oil samples are more concentrated than others, so we need to create better methods to allow us to optimize that. And moving forward, we will also need to take those chromatograms that I showed earlier and generate their mass spectra for each of the compounds and compare them to the reference library and find out exactly what those compounds are. So in total, these are the 3 projects that we worked on this summer. We studied HIPVs and their effect on bird predation. And then we genotyped chickadees and then analyzed the chemical components of preen oil.
Now, before we finish, we have some acknowledgements. First, Women for DeSales for sponsoring us. Without them, we could not do this. Mrs. Wendy Maybrook and Mrs. Carolina Graham for helping us in the lab and keeping us on track and basically planning everything for us. The DeSales Serve faculty and staff, specifically our lab instructor, Dr. H, for guiding us along the way. Dr. Joshua Slee, the DeSales University administration, Brother Dan, Father Jim, and everyone at DeSales University for supporting us every step of the way. And finally, all of the birds we have encountered throughout our research, even the ones that got away from us out of the nets. And that concludes our presentation, and we'll open the floor up for questions.
Right, nice job, ladies. Do we have any questions in-house or on Zoom? Dr. Mayville?
Yeah. Just wanted to know in the first study you guys did, why did you choose hexane as your control?
We mainly chose hexane because there was a specific study that we modeled our experiment after. Basically, we did the same thing. They used corn HIPVs, but we decided to use methyl jasmonate and methyl salicylate because that's what we were interested in. They also used hexane. We could have used, I believe, DI water. I think we were between hexane and DI water, but hexane would have been was the better solution to dissolve the rest of our components in.
Thanks.
Dr. Barnett?
Yeah, very cool. Yeah, that's awesome. So whenever you were doing the clay worm experiment, is there a way that you differentiate one bird attacking it a bunch of times versus many birds attacking it?
Yeah, that's a good question. That's why we had 3 different variables. It was sort of to account for that. we were not able to like identify which kind of bird pecked the caterpillars, because sometimes, like as you saw in like one of the previous slides, the caterpillars were absolutely just like destroyed. Some of them were just squashed into the ground, and we didn't know what to make of those. So we kind of just disregarded those in our results and noted that like in our discussion. But yeah, I think we just quantified the pecks in general as bird pecks.
Any further questions? Anything from Zoom land?
Not
Dr.
just,
Kevin?
sure, not just for these 3 researchers, but maybe for all 9. I'd be really interested to know what's next for them in terms of their careers, or I realize they're still here at DeSales, but what are you hoping for? Not only with this specific research, but with the skills and the new questions that you're finding yourselves asking, and how's that impacting the way in which your career seems to be unfolding?
Right. So I'm a PA major. So this is really important for me because I want to go into research when I become a PA, which was really interesting to me because I just wanted to do research at DeSales in general because I did it in high school and I thought it was a lot of fun and it was a lot of good experience for science, and just to sort of like get a sort of leg up in class and know a lot more that I couldn't be able to study just in class with a ton of other students. I could have a sort of one-on-one research professor to work with. But this year I found out that PAs can also continue to do medical research during their career. So this is really important for me specifically because this gives me a lot of research experience and a lot of skills that I wouldn't be able to learn just in the classroom alone or even just in lab.
And I'm pre-vet, so I really want to go to vet school, and specifically working with like wild birds. It gives me a lot of like practice, like with handling wild animals and like knowing how to handle them with care and not stressing them out too much.
Yeah, these just gave me really good, the ability to design an experiment from start to finish is something that you don't really do in class or even in lab. Normally you come into lab and they have an outline for you, and being able to create this experiment, pilot different methods and see what works and what doesn't is just really good experience to better understand the research process.
Do any of our other students care to come down and share why they joined and what they hope to get out of it or what they got out of it? You guys are all stellar students, so. Nobody run the front once.
I was leaning toward grad school for a little bit and doing this, and he said, like, yeah, I definitely want to do grad school. And I had never had like any outside background. And then after talking to Professor Bubbles, especially, she started talking about all the different things you can do. Now I'm starting to figure out how I can fit more science in my schedule and really bring those together. So now I feel like I actually have a bit more direction.
I think I could speak on behalf of both me and Needy, since we're upcoming sophomores, that we've got a little bit more experience in the courses that we'll be taking next semester. So that will be really helpful. Yeah.
Like you live on so student research have that experience in getting more science that you get. It's just helpful to understand everything, get more comfortable in all this area.
Yeah, the same as I want to go to vet school as well. And, I think specifically in our lab, we were researching viruses. So I mean, that's obviously a very medically relevant topic. So that experience alone is also really cool to see, and I'm sure I'll experience a lot more continuing education, going to vet school. So that's also really helpful.
I'm also planning on going to grad school. As Carl was saying, the integration between biology and computer science, and for really any subject in computer science is becoming so having like coding experience, that's something that I wouldn't have been exposed to without this program. So that's really beneficial.
aspect of my biology degree. I just feel like this program was really helpful for me getting in like first playing into what the field actually is like. And it's just, it feels much more involved than like any like classroom laboratory. So I'm just really grateful I got to participate in the program.
And I just want to commend you. I happen to be walking down the road, the hill, on the day when you got your white lab coat. So you look great. Very impressive. So I thought it was on the set of the ER.
And I think the students did a great job encapsulating why we created the program. It was a faculty-led initiative to create the program. It was for every reason that they said. And the other reason is that almost all of these graduate school programs, BA programs, medical school, med school, most of them require something like this now. If you want to get past that initial phase and get your application looked at, you have to do some form of research or experiential learning. So we really want to do our students correct and well by having these experiences and offering them, being able to run these programs in the future, because it really does distinguish our students from the rest of the pack. Any other questions before I turn it over to our advancement team who are going to give you some closing words? Again, I just want to thank everybody for coming, and I thank everybody for their continued support of our programs, especially the SURF program and all of our students. These 9 students are fantastic, and I cannot wait to see what the future holds for them. Because they're going to be grateful.
On behalf of the institution office and the George Government Project, we would just like to thank all of you and congratulate all of you on the great work. Another round of applause for an amazing job well done.
As I'm looking in the room, I also see a lot of parents, which I thought was really cool that a lot of the students who came here, their parents came in, whether they're alums or whatnot. Just want to thank all of our alums who are joining us on Facebook Live, obviously on Zoom or in the room with us tonight. We're really looking forward to getting back to in-person events. This is one of the first couple events that we actually can see a live room. You know, the opportunities that COVID has presented us with is this. You know, we have alums tuning in from all over the country, really, and to be able to watch what the students are doing, which is what I've heard in my couple months here as their home lab director, is their favorite thing. It's the successes that the students and faculty have. So, Dr. Slade and all the faculty here, thank you, and to the students, thank you. I know our alums are probably saying the same thing in the chat right now. You know, I think it's a credit to you of what their education of alums is seeing that you guys moving forward with the foundation they set. So thank you. Look forward to seeing you all soon. Have a good day.
Thank you.
Congratulations.